Here's some help to answer question 3 on the annotation bioinformatics 2 exercise that I created. So it says do something with the first 3,000 nucleotides. So let's see. Okay, so I wanted you to use this translating tool or translation tool to look at potential proteins in the first 3,000 nucleotides of the human chromosome 1. So the gene bank accession number is this, and I wanted to translate the first 3,000 base pairs. And I'm doing that because I really don't want you to look at the genetic code and look at all 6 translations, possible translations of the first 3,000 base pairs. So I picked a big number, mostly so that you don't feel too tempted to do the 6 translations by hand. So how am I going to do this? All right, let's see. First, I need this sequence here. I'm going to Google that and see if I can find it by that. So if I put that there, oh, look at that. I actually didn't expect it to already give me the answer. So I guess there are some updates and the tools keep getting smarter. But had it not given me the answer, here's what I was expecting to have to do. And I put the directions here. So I wanted you to find this accession. I already found it, but if I didn't find it with Google, what I really would need to do is I'm going to Google NCBI nucleotide sequence. And then you will see all sorts of things you could search on the NCBI website. So I'm going to just Google that. And here, this is what I want, a nucleotide, but I'm going to click on this so that you can see the things that are housed on this website. Here, there's this drop-down menu, and then it shows you many things you could be looking for. In our case, I want to find that nucleotide sequence, and let's see what's that number again, the accession number. I'm just going to paste it in here, and then search. And here it is. All right, if I go scroll down, this is, let's see, the Y, chromosome Y. Let's see, oh, it's the whole chromosome Y. And that picks the Y because it's a short chromosome. And this is just to give you some idea. Okay, our shortest chromosome is, I only need the first 3,000. I'm going to scroll down so that you see how much information. Can you imagine chromosome 1? It takes a while to load the page. So there you go, 42,301. Anyway, there's more than 42,301. And these guys are groups of 10. So you can count that. So 301, 11, 21, 31, 41, 51, 52, 53, 54. So 42,354. Okay, but I don't want to do the exercise in all of these. I could if I wanted to, but I just wanted to get some chunk big enough to work with, but not too big to overwhelm you. So the first 3,000, oh, these are the second 3,000. The first 3,000 is right now the part that I didn't highlight, because this one is the 3,000th first. Okay, I want just this chunk for this exercise. All right, so I highlighted that and I did the Command-C to copy it. And then I'm going to paste it in my translate tool. I already have it opened, but if I didn't, then I would go translate DNA. And because, no, who wants to remember that name? Click on that, and thank goodness I already landed on the page I wanted. If I had landed, let's see, well, there was a page with like more information. You would just look for, I think first I landed in here. I want to show you in case you get here like, oh my God, where is that? Where is that box? I just want to paste my sequence. And you're tempted to paste it here, but no. So just do a find and look for translate. Oh, thank goodness, that's where it is. So you click that, and you're just gonna go there. And oof, we get to the same page. All right, so here I pasted my first 3,000 amino acids. So let's see, 40, 50, 60, 70, 80, 91. All right, 3,000. And I'm going to leave it as the compact sequence. I could change it to verbose. So if I use compact, whenever there's a hyphen, it does not indicate indel, because remember, indel is when you're comparing one sequence and another, like, or nucleotide sequences to each other, or protein sequences to protein sequences, or at least two to each other, and one has extra things that the other sequence doesn't have, then we put a hyphen to symbolize an indel. In this case here, that hyphen is just to symbolize a stop codon. So it's a triplet of nucleotide that stops the translation. So the ribosome is reading the mRNA, and it reads H3 and says, oh, AUG, I put a methionine here, and whatever, put this other nucleotide, this other amino acid here. And then it gets to one of three possible stop codons, it stops adding amino acids. So on the verbose, it would say the word stop, and on the compact, it just puts a hyphen saying, okay, no amino acids gets put here. All right, and I'm going to use the standard genetic code. If you wanted, you could use other ones, but you're just going to do standard. And this is DNA from the Y chromosome, so we know that that's the code that the cell would use. Okay, finally, hit translate. And look how fast it did this. What it was doing is looking up these first three nucleotides, which amino acid do they encode? And then about this, the second three, for the first translation, frame of translation. How about the third three? It keeps doing that all the way to the 3,000, then goes back and skips the first, does frame 2 of translation. So these three, which amino acid do they encode? And then does this thing for the third one, and then it reverse complement. So reverse complements this, and then uses that reverse complement and says, okay, if this sequence that I had entered was a template, then the mRNA is the reverse complement of this. And then it translates the reverse complement of this. So let's see what results we got. Okay, so here are the 6 frames. Whoever coded this coded 5', 3', frame 1. Okay, good, I like that, frame 1. I don't think I like the 5', 3', but all it is saying is that it took this sequence and read, here's our 5', in the end over here's our 3'. So it took this sequence and it said, if this DNA sequence is the coding DNA, also called the sense strand, then the mRNA has the same sequence as this, except that where there is a T, there would be a U in the mRNA. Okay, so that's what these first 3 are, the ones that say 5', 3'. And then the program, whoever coded this, decided to call it 3', 5'. My goodness, how I don't like this. Nobody asked for my opinion, so it exists. But it gives you the impression that we are reading these 3, like TTA. In fact, let me include the nucleotide sequence so that you see how that's not what we're doing, because that would not make sense biologically. All right, so For the frame 1, remember it's reading 5 prime, 3 prime. There you go, that. Let me see if I can get both things in the same screen. Yeah. And then the second codon is this guy, this first 3. Okay, well, you know, this second 3 letters. And then for frame 2, it skips the first one. So in here, it has 4 letters, but that's not because it read 4. It skipped that A, and then it read AGA. And for frame 3, it has 5 letters, this first one. It just means that it skipped the first 2 and read GAA. All right, now let me show you what they called 3 prime to 5 prime frame 1. Let's go up, because I like setting my expectation before I see the result. That way I can spot either misconceptions or mistakes or flaws in an experiment, right? Okay, so if I'm going to read 5 prime to 3 prime, or 3 prime to 5 prime, so this from end to beginning, biologically doesn't make sense, but let's pretend it did. Then I would read TTA as my frame 1 for my 3 prime, 5 prime, as they called it. Or you could say, well, maybe it's a UUA. Well, whatever. So we're going to read TTA, and that would be reading 3 prime to 5 prime. Let's go down. Okay. Okay. See? It's not TTA. It's AAT. So really, it's not just reading from end to beginning. In reality, what the computer did was reverse complemented that strand. So the reverse complement of TTA is AAT. So the computer is doing the right thing. The only problem that I see with this is this name. That's, to me, it leads people to think that the computer is doing something else. But anyway, nobody asked me, as I said. So, but we can conclude that the first 3 here, the first 3 frames, there are all 5 prime to 3 prime. They are taking the sequence as given and translating that. So it's considering the DNA strand you entered is the coding strand or the sense strand, is the same thing. It has the same sequence as the mRNA. And then the other, which they call 3 to 5, is the reverse complement of this that then gets translated and then gives you those sequences. Okay, so endorent on that part. Hopefully you get the importance of getting the 5 prime, 3 prime. We always are reading 5 prime to 3 prime. It's just that where they put 3, 5, it's the reverse complement of that. And so it's still reading 5 prime to 3 prime once you reverse complement that. Okay, now the important part of this. See this part marked in red or pink? What the computer is doing is looking, where's the first methionine in this theoretical sequence? Because the methionine is the start codon. So it's saying, oh, this is probably where this protein starts. And then if that was a protein. So it's not actually looking for size, it's just looking, it's just scanning the amino acid string, and is looking for an M. And then it finds the M, it's like, oh, here. And then there's another M here, which is a methionine amino acid that is part of this protein, but not necessarily the start codon. Right now we don't know. Maybe this is the start codon and not that. This computer program is not looking for cues in the promoter, is only looking for the first methionine and how far does this sequence go until it hits a stop codon. So here, UAG, that's a stop codon. So that's that hyphen. And then it's like, oh, another methionine. Maybe this could be another protein. And then UAA, that's another stop codon. So far you'd have a protein that is, so this is your first protein here. Exclude the nucleotides. And then this is a second protein, or no, possible protein. Those are pretty short. And then, okay, computer is scanning for more methionines, finds another one. Then this one's looking better. You know, it's a little longer. And why do I think that that's better? It's longer, better. Well, we are trying to match our expectations to how long proteins generally are. So I put that information in the exercise here for you to have some idea. For bacteria, you many proteins are around 200 to 200 to 300 proteins or amino acids. We are looking at the Y chromosome, so we also have a bunch that are in the 200 to 400 amino acids. So when I'm looking at that sequence here, the output, okay, I don't need that one though. I'm looking for something that will be around that. Now that, then at least I have some idea of how long it should be. So that would be kind of short. Oh, this is looking more promising even. So here, it ends here. Okay, so I'm actually going to go back and remove the nucleotides so that I will have only protein sequence. It might become easier to compare them or to have a bird's eye view of what's going on. Okay, so that chunk's looking promising. These are all short for this frame. This is kind of long, but I think this one is the frame that gave us the longest possible protein. And this is all theoretical. We're not comparing to database. We're not looking for cues in the promoter region. That's why we don't do this for a lot of gene prediction. But I wanted to show you this tool and what it does and how to interpret it so that you do not have to go and type genetic code and look up a table when I ask you to translate some DNA sequence. So there you go, the six frame translation that the computer did for you.